taxonomic group will be the flowering plants. And I'll make it simple. We'll distinguish only between dicots and monocots. Simple enough so far, right? Don't let that fool you into thinking that the flowering plants are a simple group. There are about 250,000 species of flowering plants. They trail the insects in total biodiversity, but it is enormous biodiversity. Remember a couple of things. The older, more primitive evolutionary group are the dicots. They go back about 135 million years ago. And you're thinking, that's a long time ago. Well, compared to the evolution of plants and animals, it's very recent. The flowering plants are a young group, and they are incredibly successful. I'm going to give you a hint about their great success today, looking inside their flower parts. The dicots have floral parts in sets of 4s or 5s. I'll put a picture or two of flower parts on quiz number 9 coming back in 2 weeks. I'll mention the schedule in a minute. Now, the more recent, younger evolutionary group are the monocots. They include 3 groups: the grasses, the true palms, and the true orchids. So the monocot biodiversity is relatively simple. Everything else is in here. You see a small sampling. For example, family Brassicaceae, the mustard plants, they include your cabbage plants. Right? I've mentioned in my lecture the legumes, Fabaceae. There is tremendous biodiversity among the dicots. We're not going to go into the details of the biodiversity here in lab, but I do want you to focus on some particular aspects of pollen and ovules, something that we're going to develop. I wrote this model of a flower out for you. In my lecture, I go through developmental phases, the 4 whorls of development: the calyx, the corolla, the stamens, and the pistil. If you're in my lecture section, you know that story pretty well. You also know the story that in the development of the leaf, we start first with leaf primordia, and then given the right location, given the right hormones, we become floral tissue. Then we have pollination. Pollination is the transfer of pollen to the stigma, either in the same flower or a different flower or an entirely different plant. Pollination is followed by fertilization. Pollination is the transfer of pollen to the pistil. Fertilization only happens if the pollen tube brings a sperm nucleus to the egg, but there's more. Then, following fertilization, we have fruit development, the ovarial wall develops, and seed development. So remember that temporal sequence. And we're going to tie all these together today with your microscope slides and go over these with some detail. Now, another thing I want to do today is, you notice I still haven't finished grading the lab reports. I'm almost finished with them. I'm grading them as fast as I can. To make sure that your second lab report has, you have enough time to work on it, I've simplified the assignment. Your second lab report, you're going to use your original data set. What did you have? Data set number 3, data set number 6, whichever data set you have. You're going to use it again. Not to calculate average, length, standard deviation, pivot tables. We're not doing any of that. Now, a very simple assignment, and I'm going to show you this to you. If you've been reading your emails, you've already got a hint. I'm going to ask you to do something called, or create something called an XY scatter plot. The X is the x-axis. We call it the independent variable. The Y is the y-axis. We call it the dependent variable. And I'm going to ask you to do a simple Excel analysis called regression analysis. You may not know much about it now. In about 20 minutes, I'm going to stop you in your slide work, and I'll do a short demonstration. Did you all notice I sent you 2 emails in the last couple of days? One email has a Word document and an Excel document. The other email has a couple of links to a couple of Wikipedia articles. Those are what I want to discuss in about 20 minutes. Now, before we get there, let me take you through some details about the flowering plants. And does this look familiar? Anyone recognize what kind of flower? Some of you are flower types, aren't you? Yeah, it's a lily. And why would this be very popular in horticulture these days? Right now, all the horticulturalists that are preparing flowers, they're making big money on lilies now. You know why? This is a great time for them. Isn't the Easter season, the Christian Easter season, part of the lily story? Lilies representative of the Easter spring season. So this is a good time, money-wise, for horticulturalists that know how to get these flowers going. Is it a monocot or a dicot? Come on. How do you know? Because I said so? Flower parts coming in threes, sixes, nines, etc., etc. If they come in sets of fours or fives, dicots. Now, just to get you focused on the idea, I'm going to have a couple of photographs. They will be unambiguous, a couple of photographs on quiz number 9 of flowers. And I'll ask a simple question. Monocot or dicot? Can you count? If you can count and you can spot the parts, then it should be an easy question for you. Now, the only time it could be a tough question, what if you count 12 petals? Then you need to look at the leaf venation. I am not going to give you a flower with 12 petals, guaranteed. They will be unambiguous. Now, as far as quiz number 9 is concerned, you know next week you are not in lab. We do not meet next week. In 2 weeks, on April 20th, we're going to meet quickly for quiz number 9 for 5 minutes. I will demonstrate a second time the regression analysis, the XY scatter plot. The due date for your second lab report will be Monday, April 24th. I'm arranging that now on Canvas. Right now, Canvas says your second lab report is due on April 10th, which doesn't make any sense, and I am changing that date. Your second lab report, it's going to be a one-page report, by the way, simple to do it. Now, there are 9 quizzes. I'm only counting your 8 highest scores. If you've taken all 8 quizzes up to today and you don't want to take the 9th quiz, well, don't show up. If you don't show up, it's a 0, but that score gets dropped. Of course, you want to show up at 10:05 because you want to be here to participate in the lab. After quiz number 9, I'm going to have a simple fruit demonstration for you. I'm going to bring in fruits of all kinds that I can find, all sorts, dehiscents, legumes, any kind of fruit that I can scour up in South Florida. I'm going to bring them in and ask a simple question. Are these monocot fruits or dicot fruits? And there are some very easy ways to, very easy clues to discover. Are they aggregates? Are they poles? Are they dehiscents? And so forth and so forth. That's coming up in 2 weeks. Now in 3 weeks, on April 27th, is the second lab exam. The second lab exam covers everything since the first lab exam. The first lab exam was about 5 weeks ago. Second lab exam only covers from the algae to the fungus to the seedless plants all the way up to today. All of these slides are in the back available for you. They'll be available during open lab hours for you too. The question you might ask is, well, do I have to take the second lab exam? Remember, I count only one lab exam, the higher of your 2 scores. So if you don't show up on April 27th to take the second lab exam, you get a 0. That means I drop that score. So you rely on your first lab exam. What if you really bombed your first lab exam? No beverages on the tabletop. What if you bombed that first lab exam? Well, the second lab exam can be your savior, am I right? So if you didn't do well on the first lab exam or you missed it completely, then those slides back there are a real opportunity for you. Okay, does that catch us up to where we are now? One of the things I'm going to ask you to do is take a cross section through the base of the pistil. The pistil is the innermost of the 4 whorls. Remember, the outermost are calyx, the sepals and whatnot. The corolla, the second whorl, they are together vegetative whorls. They have nothing to do directly with reproduction. The third whorl are the stamens, and the stamens have these expansions called the anther. We're going to take a cross section through there. And finally, at the base of the 4th whorl, at the base of the pistil, we're going to get into the ovary and ovarial chambers. So take a look at the plastic model again to see where we are. We're taking a cross section through a lily pistil at the base. And again, monocot, dicot. you have three pairs of ovarian chambers, a pair here, a pair here, and a pair here. Practically all of this tissue is diploid sporophyte. We introduced that terminology with the seed plants last week. Except for right here and right here, we didn't get a good cross-section of it here. This is a structure called the embryo sac. The embryo sac is surrounded by ovarial tissue, but the embryo sac itself contains gametophytes. Gametophytes, fancy word for haploid cells. You remember? The egg for fertilization is inside that embryo sac, but there's more detail. It gets a little busy inside. Once you're inside, if you have this is a blurry photograph, the embryo sac, you might find the 8 famous haploid cells that are inside. One of them is the egg. The other 7 are interesting, and they're going to be an interesting story for you to look at. Not so much the lab, but I want you to focus on the story much more when I get to it. Inside the embryo sac, this is all diploid sporophyte tissue, but this holds the gametophytes. The embryo sac is the container for the gametophytes. As I said, there are 8 identical haploid nuclei in here. One of them will become the egg. You probably won't get a good slice enough to find this. What about the other 7? I'm going to show you what the other 7 are involved with in a minute. The anther, by contrast, is going to be a little simpler to deal with. When the anther is mature, it dries and cracks open. This anther is now dried and cracked open. These are the gametophyte, the pollen grains. Now, interestingly enough, the pollen grain actually contains 3 nuclei, 3 haploid nuclei. So it's not a single sperm nucleus, but actually 3 of them. They have an interesting development too. It gets a little busy with the pollen grains. And the trick is, the pollen grains have to be delivered from the anthers to a pistil somewhere. Maybe a pistil in the same flower for self-fertilization, like Mendel's bees, or a distant flower. A pollinator might be involved, or wind dispersal might be involved. All of this is diploid sporophyte tissue. The gametophyte tissue, the haploid gametophyte tissue, they are represented by the pollen grains. On page 432, 433, I'm not sure if your edition of the lecture has the same pages, but you'll find it soon enough. Make sure you take a look at this particular figure. Look at your index for the life cycle of the flowering plant. Because now we're at the pinnacle of the seed plants. Remember, we were dealing with seedless plants, like mosses and liverworts. The gametophyte stage was the larger, longer-lasting, more dominant life stage, the haploid stage. The sporophyte stage of mosses and liverworts is actually tiny, microscopic, very short-lived. With the seed plants, we're going in the other direction. Now the sporophyte stage is the dominant life stage, and the gametophyte stage is very short and brief and microscopic. Well, when I say brief, the pollen grain contains the sperm nuclei, and the pollen grain itself can be long-lasting, but the vast majority, 99.99999% of the pollen that a plant produces, it's not going to land anywhere where it can have an opportunity for fertilization. 1 out of a million, like a lottery ticket, is what we're talking about with pollen grains. I'm going to go into a lot more detail about the life cycle here in lecture, but I want to remind you something about meiosis. Do you remember meiosis? We're going to review it in lecture next week. Meiosis means that you take a diploid nucleus, 2 chromosome sets, one from the mother, one from the father. You take that diploid nucleus, you divide it twice, and have 4 haploid nuclei. I hope you remember that much about meiosis. Now, does that occur in the pistil? Here's our embryo sac. Before fertilization, we have a single diploid cell. It's the germ cell. It undergoes meiosis to produce 4 haploid nuclei. They're all different. Here's where it gets interesting. This happens only in flowering plants. 3 of the haploids degenerate only one remains. So you eliminate 75% of the meiotic products very quickly. That one nucleus will divide mitotically once for 2 daughter cells, twice for 4 daughter cells, 3 times for 8 daughter cells. Does that sound familiar? Didn't I tell you the embryo sac has 8 identical haploid nuclei? Yes, they do. One of them is actually the egg. It finds itself down here. These migrate around. This is the egg. 2 in the middle fuse together, and they will be fertilized by a second pollen nucleus. So let's bring the pollen grains in. The pollen grains have a different maturation process. What they do is they will pack 3 haploid nuclei into the pollen grain, and when pollination is successful, a pollen tube develops. One pollen sperm nucleus fuses with the egg to create a diploid zygote. You know that story. The second sperm pollen nucleus fuses with this central pair of haploids to create a triploid cell. A triploid cell is 3 chromosome sets. 1 from the pollen, 2 from the female ovule. That becomes something called triploid endosperm. I've told you about endosperm very vaguely so far. Endosperm is what the cotyledon stores a lot of. The cotyledon or cotyledons are filled with endosperm. It's tissue that is triploid, and it contains a lot of nutrient value. All of the proteins and lipids and fats and nucleic acids and carbohydrates and starches that the plant will need, the embryo will need, is made up in the endosperm, triploid tissue. This process, by the way, results in something called double fertilization. You use 2 sperm nuclei, one to fertilize the egg to make a diploid zygote, and then an embryo, and the second to fuse with this central pair of haploids to create triploid endosperm. This doesn't happen in any other plant group, only in the flowering plants, not in the conifers, not in the cone-bearing plants, not in the ferns. None of the other plants do this. It makes the flowering plants unique in a lot of ways. It also makes them tremendously successful. You'll find out why they're so successful and why double fertilization has been so remarkable in evolutionary history. We'll get into that story in the lecture. So what I would like you to do for the next 15 minutes or so, I want to show you some things on the slides to watch for. They're simple to observe, but put some thought into this. What part of the plant are you looking at? And I left this plastic model up here for a reason. I want you to take a look at it and make sure you know where the material is coming from that you're looking at. The first slide is the, as I find it, the cross section through the anther. And I want you to visualize what exactly is the tissue that you cut into. The tip of the anther up here. It's the third whorl, the top, the expanded portion of the stamen. It's filled with pollen grains. Each pollen grain has 3 haploid nuclei. Some other things to watch here. Notice that the anther itself has a lot of diploid tissue. This is all diploid tissue. There's the stamen stalk. This is the expanded part of the anther. These are the gametophytes. The haploid gametophytes are the pollen grains. These are diploid sporophyte tissues. So keep that in mind. The adult tissue, the sporophyte diploid adult tissue, surrounds the haploid gametophytes. The gametophytes are released when that anther dries and splits open, and they're released, and then they can be distributed either wind pollination, if that's the fashion, but a lot of flowering plants are animal pollinated. The bees, the hummingbirds, the butterflies, whatever the pollinator is. The next slide I want you to look at is going to be labeled either lily ovary, or it might be labeled lily embryo sac. And here we are. Monocot or dicot? Three sections, right? And we're cutting right across the base and cross section here. Notice the vast majority of tissue in here is diploid sporophyte adult tissue, the adult plant. This is the embryo sac. This is all diploid sporophyte tissue until you get into the embryo sac. Not all the cuts are going to get you into the embryo sac. That is an embryo sac. And you might be lucky and you might even find a couple of haploid nuclei in there. There are 8 of them, but you'd have to be very lucky to get all 8 of them in an individual slice. The gametophytes, very, very, very few in number, and they are tucked inside the embryo sac. The embryo sac is surrounded by diploid adult tissue. Remember, in fruit development, something we'll talk about in a couple of weeks, the ovary wall produces the fruit in the maturation, but only after pollination and fertilization has taken place. Without the fertilization, fruit development is practically impossible. Unless you're dealing with seedless fruits, and that's a different issue. Okay, is that enough for you? I'm going to give you about 10 or 15 minutes to get organized on these slides, and I'll stop you and take you through the XY scatter plot.